welcome to Educon. In this session, we'll learn about living state and also enzyme and their activity. Coming to living state, maintaining of biomolecules at constant concentration of molecules, ions in the cell is very important for its survival. Yes, certain molecules and ions are very much important for the cell to keep it in living state. So those molecules, ions should be there at constant concentration, so the cell can survive. And also biomolecules are present at different concentration. Okay, it is not, for example, blood concentration of glucose is in normal healthy individual is 4.2 millimolar to 6.1 millimolar, while that of hormones it will be in nanograms. Remember this. Okay, these are the standards you must remember, because you may have a direct question like what is the concentration of glucose in blood in normal healthy individual. So this line is important for you can answer with this. Okay, you must remember all this. In chemical and physical reaction process, in chemical and physical reactions, process moves spontaneously to equilibrium. That is, as we know, in chemistry you might have learned. So in chemical and any reactions, the process moves spontaneously to equilibrium. In living systems, the living state is non-equilibrium and ready to perform to work. Okay, this is also important line. 1, 2, these two lines are very important. Okay, in living system, the living state is non-equilibrium and ready to perform work. Okay, and in living systems, non-equilibrium state is achieved by energy input. So how the cells maintain non-equilibrium state? Okay, because it is achieved by energy which is obtained through metabolism. Metabolism means catabolism and also anabolism. So the non-equilibrium state is achieved by energy input, which is in turn provided by metabolism. Hence, living state and metabolism are synonymous. This point is also important. Okay, without metabolism there cannot be living state. Okay, so here we have 4 to 5 lines to be remembered, and we need to work on these things by seeing the questions in previous NEET or any you will be having a lot of free exercises available on Google, so you can search and definitely you can learn a lot from these points which we discussed just now. So where the glucose concentration of glucose in blood for normal healthy individual, and also in living systems is a non-equilibrium state. Okay, and it is living state and also metabolism is synonymous. Okay, coming to enzymes, almost all enzymes are proteins. Okay, all enzymes are proteins. Nucleic acids which behaves like enzymes are called ribosomes. Okay, nucleic acids are different, okay, but sometimes they behave like enzymes, so they are called as ribosome. Enzyme, like any protein, has a secondary and tertiary structure. As we know, proteins, they are having 4 different types of structure. One is linear, another is beta sheet and alpha sheet, then tertiary and quaternary. So same like enzyme, same like protein, enzymes do have secondary and tertiary structure. When proteins fold up on itself, it forms many crevices and pockets. Such pockets are called active site, for example. So this is the tertiary structure. So we are having a bend here. So these bends, it will form a crevices. So here. So these crevices are actually called active site. Okay, which catalyze the reaction at higher rate. At active site of the enzyme, substrate conversion to product takes place, where the substrate will convert into product. That we'll see through the pictorial representation in the next slide. Enzyme catalyst differs from inorganic catalyst, as the latter works efficiently at high temperature and high pressure. Inorganic catalyst means manganese, lot of mineral ions like manganese, magnesium, sometimes iron, different. Okay, so enzyme they are the biomolecules. Okay, they are the proteins. Proteins are denatured at higher temperature and high pressure. So enzyme catalysts differ from inorganic catalysts. As the inorganic catalysts, they work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature, that is above 40 degrees. But at the same time, there are certain exceptions, like certain enzymes isolated from organism, which normally live under extreme high temperature, such as hot vents and sulfur spill, are stable and retain their catalytic power. There is exception. We have certain enzymes, okay, where which are isolated from organism, which normally lives in under extremely high temperature, and like hot winds, sulfur springs, and also they are stable and retain their catalytic power. Chemical reactions. Chemical compounds undergo two types of change. Okay, what is chemical reaction first? So from this thing to that thing. So for example, a physical change and also a chemical change. Now we have a pictorial representation here. Okay, we see in chemical change we have combustion, rotting, rusting, digestion. Okay, here in rotting, okay, the microorganisms, they break down the larger molecules into smaller molecules. Okay, same in digestion also. Here also in rusting same, in combustion too same. But in melting, physical change, melting, shredding, boiling, chopping. So melting, what is happening? So it is turning into liquid, from solid to liquid. Okay, and in chemical change, shape of the molecules along change in shape of the molecules with breaking bond. Okay, here change in shape of molecule along with breaking bonds. Whereas in physical change, in chemical change, bonds are broken and new bonds are formed. Whereas in physical, shape of the molecule changes along with the breaking bond. So in chemical compounds undergo two types of change. A physical change, that is change in shape of molecules with breaking bond. This one here, what is happening? The ice cream is actually a solid before, because the water it is under freezing condition. So what happened when you take it into a higher temperature? It will start melting. So here physical change happens. So it turns from solid to liquid. Same like in shredding, a large paper will turn into small pieces, and chopping also, when you cut tomato or any vegetable, you are changing the shape of the fruit here. And in chemical, bonds broken and new bonds are formed here. Okay, here we have a barium hydroxide, BaOH, and hydrogen sulfuric acid, H2SO4. So here what is happening? You can see sulfate is joining with the barium and the release of water. So here this is a chemical reaction where new bonds are formed and the chemicals are broken. This is one of the example for this. Rate of physical or chemical process refers to the amount of product per unit time. Rate of physical or chemical process refers to the amount of product per unit time. This is also important line. Remember this, and this equation you need to remember. Okay, it is refers to the amount of product per unit time. Product, this is the product, and this time. Rates of physical and chemical processes are influenced by temperature, and rate doubles or decreases by half for every 10 degrees change in either direction. Okay, for example, it is increasing, that we just learned in previous slide that at certain that we'll discuss how exactly the enzymatic reactions are affected by temperature, pH, other factors. So here rate doubles or decreases, means if 10 degrees it is there, if it is 20, the reaction rate is double. If again it comes to 1, the reaction rate falls to half. Okay, Catalyzed reaction process proceed at rates vastly higher than that of uncatalyzed reaction. For example, this here we will discuss that here, okay, where here carbonic anhydrase is the enzyme, okay. Now CO2 plus H2O. So what is the result? Carbonic acid. Fine. So in the absence of enzyme above reaction, so for example, if there is no any carbonic anhydrase here, the reaction will be very slow and produce only 200 molecules of carbonic acid, that is H2CO3. And in the presence of enzyme, carbonic anhydrase, the reaction speed is 600,000 molecules per second. See the difference. So the reaction rates are vastly higher when there is there is enzyme involved in the specific reaction. Reactions can be multi-steps, okay, it can be multi-step or in single step. So reaction can be multi-step where each reaction can be catalyzed by same enzyme, okay, or different enzyme complex. Okay, fine. So from chemical reaction we saw what is chemical change, what is physical change. A chemical change is where the bonds are broken, new bonds are formed. Where the physical change, bonds are broken along with the change in the shape of the molecule. Okay, and we have to remember these points. Rate of physical and chemical process refers to the amount of product per unit time. Okay. Enzymes in reaction. How exactly enzymes works? So we already know that enzymes are almost all enzymes are proteins. So enzymes are proteins with active site where substrate is converted into product. So here this is the substrate and this is the product. Okay. Now we have a picture here, catalytic cycle of an enzyme. So and here this is the active site. So this is the substrate where it comes in contact with the active sites of the enzyme, forming enzyme substrate complex. This complex formation is transient phenomenon. This is the enzyme substrate, okay, and here enzyme product complex. Okay, during this state where substrate is bound to enzyme active site, a new structure of the substrate called transient state structure is formed. Okay, this is the temporary structure which is forming during reaction. Once the bond making or breaking is completed, the product is released from the active site. Okay, once the old bonds are broken and the new bonds are formed, the product is released from the active site. And this we can see here a small representative. This is the enzyme and substrate. And then at first enzyme substrate complex will be formed, which is temporary phase. Again enzyme product phase. This is also temporary. At the end, enzyme is released. Again, go for go into the another reactions, whereas a product is released. Okay, and this ES, that is enzyme substrate complex, and EP, enzyme product complex, they are short-lived. That is transient, that is temporary. Okay. Factors affecting enzyme activity. Enzyme activity can be affected by a change in conditions which can alter the tertiary structure of the protein. Okay, enzyme activity, as I told you that enzyme, it is almost all enzymes are proteins. Like they have a secondary and tertiary structures. Okay, when enzymes they fold upon itself, it forms cleavages that we discussed in previous slide. So in this, the activity at the active site can be altered or changed under certain condition, and it alter the tertiary structure of the protein. So enzyme activity can be affected by change in the conditions which can alter the tertiary structure of the protein. What are the conditions? Number one, temperature, pH, change in substrate concentration, binding of specific chemicals which regulates its activity. All these four we will discuss one by one. Activity of the enzyme is also sensitive to specific chemicals that binds to enzyme site. What is enzyme site? That is the active site which is present on enzyme. So activity of the enzyme is also very sensitive, sometimes to certain chemicals, okay, which binds to active site. Then the catalyzing power of the enzyme will be reduced. When binding of chemical stuff, when the binding of chemical shuts off enzyme activity, the process is called inhibition. For example, the enzyme has to perform certain function, certain reaction. This what happened? Certain chemical stuffs which takes place on the active sites of the enzyme. So then the process is called inhibition, whereas the chemical involved is called inhibitor. So if the inhibitor closely resembles the substrate, for example, if it is appearing same like a substrate in its molecular structure, then it is termed as competitive inhibitor. Okay, and competitive inhibitors are used in the control of bacterial pathogen. This is important line, okay, remember this. And whatever you see in the video, try to, after listening to this session, go and see into the NCERT book and study from it. So the more you revise and you remember all these things, okay. Competitor inhibitors are used in control of bacterial pathogens. So here we have around 3 graphs, okay, how the enzyme enzyme activity is affected at different conditions. First is temperature. Now in the above graph, one can observe the enzyme activities high at particular temperature, and activity decline both below and higher than optimum. So here this is the activity falling, and here also there is no activity. But only at a certain temperature the activity of the enzyme is fully restored. The beyond this, at lower temperature or higher temperature, the proteins are, because most enzymes are protein, they are denatured by heat. Coming to second graph, here how the pH effect. In above graph, one can observe the enzyme activity at optimum pH, that is 8, okay, and activity decline both below and higher than pH level. So this is the correct optimum pH required for the enzymatic activity. If it is 7, 6, 5, 4, then the activity will be reduced. Same into higher pH level. Same for the higher pH level. Coming here, substrate concentration are also reaction. Increase in substrate concentration initially increases the speed of the reaction. Okay, for example, we have enzyme and you keep on adding the substrate. Okay, and the substrate will go and stick to the active sites of the enzyme. Okay, then as far as the enzyme is present in that, the reaction speed increases because we are adding the substrate. There is enzyme in that. Reaction reaches a maximum velocity and does not exceed as enzymes are saturated. Once all the enzymes which is present in the system, if they are preoccupied by the substrate, even if you add more substrate to the solution, that reaction will be constant. It will not move because the enzymes are already saturated. They have enough substrate to convert into product. Okay.